Alright guys, 13.1 guided notes. Before we start, just going to say be good for the sub. Even though this is just a recording, I know that you guys need to be good for the sub. Otherwise this class will get miserable. Remember what I said last time. Here we go. Talking about RNA. RNA is ribonucleic acid, which is different than DNA, which is deoxyribonucleic acid. RNA has nucleotides that are a little bit different. Pretty much the same though. So instead of a deoxyribose sugar, it has just a ribose sugar. It also has a phosphate, just like DNA, and it has nitrogenous bases like DNA. So RNA, remember, it's a nucleic acid just like DNA, hence the nucleic acid part of that word. So it's pretty much the same structure, but it is a little bit different, and it has different functions than DNA. So biggest difference is that the nitrogenous bases for RNA are a little bit different. There's no thymine, there's no T, which means that thymine is replaced by uracil. You guys who did the lab, you saw that there was those white straws labeled uracil, and that's what those were for making RNA, and we will probably be making RNA with that lab, so you guys can see how that is structured. Uracil is complementary to A, because there's no T, your A needs a partner, U does that. So instead of A, T, C, and G in DNA, RNA has A, U, C, and G. RNA is single-stranded. So while DNA was a double helix, RNA is just a helix. It comes in 3 different types. You got mRNA, rRNA, and tRNA, and we're going to talk about all 3 of those. Functions of RNA, the first one is messenger RNA. It works as a messenger. Look at that. It also carries a copy of the gene from the DNA in the nucleus to a ribosome in the cytoplasm. So we have all this DNA in the nucleus, and then we need RNA to actually go in there, get made right next to it, and then that actually will move out of the DNA. Oh, dang it. Thought I was going to be smooth there. So that RNA gets made in the nucleus, and then it actually makes its way out of the nucleus so that it can do what it needs to do out there. Okay? It holds the instructions for the protein that the gene codes for. So remember, genes are portions of DNA that code for a protein. DNA is the instructions for a protein. But it can't leave the nucleus. The ribosomes make the protein, so it takes the instructions, mRNA takes the instructions from the nucleus to the ribosome so the protein can actually be made. Okay? Alright, moving on. Actually, this is what it looks like. Notice that this is mRNA, it's single-stranded. It has a bunch of base pairs, just like a piece of DNA, but it's single-stranded. They aren't connected to anything. So next we got rRNA, ribosomal RNA. This makes up the majority of the ribosome. 2 pieces, large and small. We talked about this a long time ago. So hopefully we can remember a little bit that the ribosome has 2 pieces. It makes proteins, and it's made mostly of ribosomal RNA. So if you guys look here, those are the 2 subunits, mostly made of ribosomal RNA. It's also got some proteins in there to help it function. Okay, moving down here, tRNA is transfer RNA, and I think of this as like the hitman of RNA, because it goes and gets the amino acids that the ribosome needs. So a ribosome says, hey, I need this amino acid, and the transfer RNA goes and gets it, okay? So it can be incorporated into the protein. Remember, amino acids are pieces of protein. So when you guys are taking your protein shakes to get big, to build your muscle, you're actually consuming a bunch of amino acids, and your body breaks them down so that your ribosomes can put them back together in the order that you want. So you want the right amino acids for adding protein and building muscle. And the tRNA goes and gets those from the areas of your cell and brings them to the ribosome. So it's a hitman. It goes and gets what it needs. Okay, this is what it looks like. It's got a funky shape. You can see that there's different parts of it that pairs up with itself, so it makes this folded shape. Other parts called the anticodon, that is when it doesn't pair with itself, so it kind of wraps around. But that's basically what tRNA looks like. It's really interesting. People spend a lot of time studying this, trying to figure out how it works and how it does what it does. So first we got to talk about RNA synthesis, also known as transcription. Remember that word, transcription. Okay? Transcription is actually, it means what it is. When you transcribe something, you are basically just writing it again. So if I give you a sentence and I ask you to transcribe it, you are just going to write it on your own piece of paper. You're not going to change anything about it. So that's what happens with mRNA with the DNA. It takes the information that the DNA has, transcribes it onto RNA. Same information, same kind of information. It's a nucleic acid information. This happens in the nucleus, and it requires RNA polymerase. I apologize for my bad notes. That is RNA, not DNA. It does all these things. It finds the promoter region, it binds to the DNA, it opens up the DNA strand, it adds on complementary RNA nucleotides, and releases the finished mRNA. Now this is not my second take at this, so let me erase this quick. So we got a piece of DNA with its different bases on there. RNA polymerase just goes and says, hey, we need this part, we need this part, we need that, matches it up. Remember, it's matching wherever, say, DNA has an A, that's where the RNA needs a U. And wherever the DNA has a T, that's where the RNA needs an A. And G needs C. So it's the same deal. It's the same language. It's just being moved from, or being transcribed from DNA to RNA. But the RNA, that's not the final process. The RNA must be processed, edited before it is, or after it is synthesized. So after you do all this copying with the RNA polymerase, then it actually has to be edited to make sure that it is right. Okay? So we're going to talk about introns and exons. Introns are intragenes. Intra means in between. They're segments of DNA that are inserted into the gene. It's extra information. It's stuff you don't really need. This interrupts the function of a gene, and it must be removed before translation, which is protein synthesis. This word translation. So we talked about transcription is just copying something down exactly, just on your own piece of paper. Translation would be like translating from one language to another. So you would translate my sentence from English to, say, Spanish. And that would be translating something. So we're translating this information from being a nucleic acid form of information to a protein form of information. So, but that doesn't happen with introns, it happens with exons. The X in exons means expressed. Don't think that exons get cut out, they don't. You want to keep exons because they're expressed in the gene. They're the segments of the DNA that are the instructions for the gene. All that remains in a processed RNA are the exons. They can leave the nucleus and go to the ribosome. It's the final form of the mRNA. So we look here, the introns, those are the not necessary parts. The exons are necessary. You want exons. So the final product is all exons. Guys, I know I talked really fast, I moved fast. I'm a little bit in a time crunch making this, but you guys can pause, redo this. I will post this video to Canvas under the guided notes so you can watch this at home. Those of you who are sick, you can watch this at home too. Hopefully you found this, and hopefully this helps you. We will talk more when I get back next week. Remember to do your vocab and your section worksheet. You have the rest of class to do that. Be good for the sub. Don't get a bad report or this class will be miserable. Okay? I will see you all next week.